recording. Good morning, grade 9, my advisory. We are now in the continuation of our discussion. Okay, this is again part of genetics. We still stuck in codominance. Again, codominance is a form of dominance wherein two alleles are equally strong and they are both visible in the hybrid genotype. Okay, again, genotype is pertaining to set of genes. Okay, for example, when a white chicken is crossed with a black chicken, the result is not gray. Okay, if it is gray, so tendency this is what? Incomplete dominance, diba? If the result is gray, it is incomplete dominance because what? Because neither of two parents or neither of the two colors resulted in the progeny. Oh, again, incomplete dominance, we're talking about incomplete dominance. Neither of the set of parents is actually projected on the progeny. There is no the same color with the progeny. Instead, it created another variety. This is variation already. Another variety, meaning to say that in the offspring, you cannot see red. Okay? You cannot see also white. This is in Gumamela. Why? Because this is incomplete dominance. The cross between two different alleles will result to another form of allele. Okay, this is heterozygous. Another form is when the set of alleles are equally strong and they are visible in the hybrid genotype. So, like for example, if we cross a white chicken and a black chicken, the result is not gray. Why? But a chicken with both black and white, because the two alleles are equally strong, they manifested on the progeny, just like this one. Okay, we cross white and black chicken and the result is checkered. So you can actually observe the black and the white in the progeny. If you observe the white and the black in the progeny, which the parents are actually given in the parents, or the parents is carrying it, it is codominance. Did you get the point? Can you see the comparison now?
Yes.
Okay, if it is
Yes.
complete dominance, you cannot actually see white and black in the progeny. You can only observe gray. Okay, gray or other colors. Okay, which is not actually found, or which the parents don't have, or which the genes of the parents don't have, or do not contain. But in this case, in codominance, you can observe that the progeny has black and has white because they are equally strong. Okay, it actually manifested in offspring. You can observe it in the offspring. That's codominance, right? Since crosses involving traits that show incomplete dominance and codominance generate the same genotypic and phenotypic ratios, yes, we can observe that the genotypes and the phenotypic ratios are the same in the progeny, simply because they created all what? They created heterozygous all. Okay, the progenies there are heterozygous. In the progeny, it is often difficult to distinguish these two types of inheritance. Okay, it is really difficult. But because of the colors, okay, because of their characteristic, we can identify. But it is really hard. Multiple alleles, okay? Multiple allele is type of a non-Mendelian inheritance. It is not actually pattern in Mendelian, okay, like dominance and recessiveness. That involve more than two alleles, okay? Multiple alleles exist when there are many variations. Okay, an example of this is the human blood type. Remember the ABO system? The blood type A, B, and O. Okay, this is actually part of the multiple alleles. Epistasis. Okay, the interaction between two or more genes in control of a single phenotype results in epistasis. Okay, Epistasis occurs when the action of one gene is modified by one or several other genes, which are sometimes called modifier genes. Let's take a look at this one later. Okay, a gene can either be masked or modify the phenotype controlled by other gene. Okay, it is actually covered up. Masking of epistasis occurs when a gene of one locus masks the expression of the gene of the second locus. Hence, the phenotype of the gene at the second locus is not expressed. On the other hand, the modifying epistasis occurs when the gene of one locus modifies or changes the expression of the phenotype of the gene of the second locus. The gene that does not masking or modifying is called epistatic, while the gene that is masked or modified is called hypostatic. Okay, or hypostatic. Right. So what is epistasis, by the way? Okay, when you're talking about epistasis, this is now the interaction of different genes or different alleles. Okay, we are actually composed of different genes, right? Human itself has different genes for your hair, different genes for your eyes, the color of your eyes, facial expression. This is the reason why, okay, take note, this is the reason why among all your siblings, okay, there are some siblings that tend, that is not actually, you're thinking about not belonging to a group. Okay, asa man ni siya naliwat. They are not, one of the siblings or two are not actually belonging to a group. All of your siblings are white. Okay, you are all white, and the other sibling is black. Okay, this is the reason behind. That is because of epistasis. Might be that one gene or the gene for the white is covered up by another gene. It is actually modified. That is the modifier gene. What else? Masking gene. Here, or masking epistasis. The gene of the one locus is being masked, okay, with the expression of the second locus. Like for example, if you have a black hair, all of you black hair, okay, but there are other one, okay, that is blondie hair in your family. Okay, it is because of the masking epistasis. It is actually the first gene on locus masks the expression of the second gene. Okay, the black. So tendency is, the result is what? Blondie hair. Those are just few. Okay, that's how difficult the prediction in genetics is. But of course, because of technology, we can usually identify. Okay, even the set of genes that you have, we can identify them through a single cell. Okay, by genetic engineering. Okay, we can identify the set of genes that you have, and of course, predict the result. You can always also change a specific gene or locus or loci. An example of masking epistasis is observed in a coat color of Labrador retrievers. This is really common. Okay, Labrador retrievers, dog. Okay, a breed, a dog. Masking epistasis is observed when a color varies from yellow to black and is controlled by Labrador retrievers coat color. Okay, this one, oh. We cannot really identify the progeny of a dog even if it is a pure breed. Okay, sure, we had just have a mate between a black Labrador and a yellow Labrador. Okay, but the progeny has white Labrador. How would that be possible? This is because of the masking epistasis. That is the reason behind. That is why usually dog breeders choose the best and choose the unique ones. Those are unique. Those are also expensive. What else? The Chow Chow, the coat of the Chow Chow. Are you familiar with Chow Chow? The breed Chow Chow. It looks like a bear.
Not really.
Yes.
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 Bear, correct? Okay. The coat of the Chow Chow will actually differ on each other. Before, we have only Chow Chow that is colored yellow and colored um yellowish and orange, I think. That's their coat originally. But now we can observe Chow Chow with black, Chow Chow with white, okay, white coat. How would that be possible? It's a different color now already. It is not it does not belong to any of the original Chow Chow. That's the reason behind. And I saw also Chow Chow that is gray color. Okay, the coat is gray. Even the Siberian Huskies. Okay, this is common in Siberian Huskies. The color of their eyes. Okay, how would it be possible? Okay, those are the unique characteristic, and the more unique they are, the more expensive they are. That's the reason behind. Two dominant alleles, one for the presence of dark pigment and another for the degree of pigmentation. B, if the Labrador Retriever's genotype is EEBB, okay, if EE, all capital, meaning to say that it is dark and the pigment is light, okay, because this is the pigmentation. So, or EB, meaning to say this is heterozygous. By the way, this is homozygous and homozygous, and heterozygous and homozygous. The dog's fur will be chocolate brown. See? Because there is a light pigmentation there and a dark pigmentation. So the result is chocolate brown. Okay, this is common in Labrador Retriever, right? And this is really expensive also, the chocolate brown. They're looking for that in the Golden Retriever. And the nose itself, okay, the form of the nose, they also want those unique formation of the nose. If the genotype is EEBB, meaning light, okay, light, pigment, and light, degree pigmentation is very light, or EEBB, or EEB, capital letter B, the 4 will be yellow. Although the B allele in dominant is its expression will be masked by an E allele. So those are just few masking, no? It covers up the expression of the second allele. So sex-linked traits. Do you have any questions so far? Any experiences? Like you experienced that your dog really gave birth to the different progeny, and then you
My
observe
cat,
that
sir.
yes, your cat, it has a different
Yes.
progeny.
It's for children, sir.
Yes.
The kittens. Yes. Um, if I remember, sir, the father was orange,
Mm-hmm.
and then three of the kittens are orange, and two of them are like the mother, white, but also with orange.
Mm. See, that is actually masking, right? Let's move now to sex-linked traits. Okay. Scientists discovered that there are some traits or genes that are linked to sexuality or linked to a particular sex. It could be female and male. So what are those? How do they interact? How can we affected with those sex-linked traits? American um, geneticists Thomas Hunt Morgan studied the genetic variation of fruit flies. Okay, fruit flies is your Drosophila melanogaster. Drosophila melanogaster is common at home if you have actually banana. These are flies. These are small flies that actually living on those rotten fruits like bananas. Okay, they're really, we can actually observe them if you have banana at home. Okay, they will just come out of somewhere, and these fruit flies can actually breed faster. So we can observe faster, we can have the result faster in this Drosophila melanogaster. In 1910, he observed one white-eyed fly among a large number of red-eyed flies. Okay, Morgan bred the white-eyed fly with red-eyed female. Okay, all of the offspring in the first generation. Okay, first generation or F1 had red eyes. He then mated flies with this generation. So all red-eyed flies na iyang gimig. Okay, the second generation of the offspring, as a result, 3/4 of the second generation of the offspring had red eyes, and 1/4 had white eyes. So this is 3:4. 3:1 rather. 3:1. These results seemingly confirm with the result that Mendel observed in his garden piece. One striking finding is that all the white-eyed flies were males. Meaning to say, this is linked to a particular sex, or this is sex link. Okay, hemophilia. Okay, hemophilia or hemophilic breeder's disease is a sex-linked inherited disease in which the person's blood does not clot easily. Okay, resulting in persistent bleeding. Hemophilia is a recessive inherited disorder caused by defective gene on X. Meaning to say, this can only be inherited in what? Both male and both female. Why cher? Because male is XY and female is XX. Okay? Males have an X and Y chromosome combination, and the Y chromosome comes from the father, while X, okay, is a chromosome which comes from the mother. But this is generally because female is X and Y, usually this will be carried by the XX or female. So usually the hemophilic organism is usually female. This is carried by female. Okay. Colorblindness is a sex-linked trait also that affects males much more often than females. Okay, because this is linked to XY or the Y chromosome. Colorblindness in males have difficulty in distinguishing red and green. The ability to differentiate two is controlled by a gene located in the chromosome. The inability to distinguish colors is due to the recessive allele of this gene. Okay, this is actually carried by a Y chromosome, which can be found in males. Okay, sex-influenced traits. Sex-influenced traits are autosomal traits that are influenced by sex. An example of a sex-linked trait is the baldness pattern. Okay, baldness in male. You can observe the baldness. Okay, you can actually observe them in male rather than females. Okay, why is that so? The baldness gene is found only in the X chromosome, since males only have one X chromosome. They have the higher chance of being bald than females. Okay, baldness is influenced by hormones such as testosterone and dihydrotestosterone. Okay, however, high levels of stress may also trigger the expression of this gene in females. That is why there are some females, okay, experiencing baldness. This is because of the external factor rather than the genetic factor. Okay, external factor, which means stress. What else? The usage of chemicals, like for example, the shampoo, allergic reactions. So this can cause also baldness. Okay. Sex-limited traits. Sex-limited traits are autosomal traits and found only in one sex. An example of this is the bright plumage of a certain male birds. Okay, this trait does not appear in females of the same species. Male chicken develop large comb while female chicken of the same breed or bred do not. Okay, so if it is large, it is male. If it is small, it is female. Okay, milk production is found in cows but not manifested in bulls. In humans, a male child may inherit his bird growing trait from his mother, although his mother does not show it. Okay, because again, this is sex limited traits. There are some characteristics that can only be found on males and can only be found in females. Okay, these are the sex limited traits. Breeding strategies. Okay, do you have any questions so far? Are we too fast?
None, sir.
All right. If you don't,
None.
if you have question, please raise it directly so that we can um I can assist you with your concern. I can answer your questions. Right? I really appreciate students that are asking questions. In my case, if other teachers don't want their student to ask questions, in my case, I want my students to ask questions. In that certain degree, I can identify whether you are actually listening or you are actually learning out of the discussion, right? Breeding strategies. These are the strategies being used in order for us to choose the best. Remember that when you breed an organism, you are thinking about making the best. This is the reason behind breeders don't want to breed their dogs, for example, with the other variety of dog. Why? It's a big no-no. Sure. Why sure? They don't want to breed it with, like for example, um, a Siberian Husky. You don't want to breed it with um chow chow to create chowski. Okay, I heard a lot about it. Okay, why? Because you're destroying the breed. You're not preserving the breed. Breeding is preserving the breed and choosing the best. This is the reason behind. Okay, selecting parents with desirable traits is the oldest method of getting better traits in offspring. By constantly breeding such parents, the breeder develops a pure breed or a thoroughbred types. Okay, this is the reason also it is controlled by um a card. Okay, if you have a pure breed dog, you will have a I think PCI ba to? Okay, the PCCI the card, the license. Okay, correct. It's a PCCI, Philippine Canine Club Incorporated. So you will be given a card from PCCI, which entails or validates that your dog is a pure breed. This is to prevent crossbreeding, right? Pure breeds carry two dominant genes or two recessive genes for a given trait. Okay, a farmer may utilize the same technique, crop yield, that is to make crops or a pest resistant to yield more products that are bigger and sweeter, or to eliminate undesirable crops. This is actually happening now. Okay, the breeders of the crops, like for example, corn, you can actually see that a lot of really good grown corns appears in the market. Okay, these corns are pest resistant, meaning to say it can actually resist pests. And aside from that, this corn also is really good in taste, in terms of taste, meaning it creates a lot of sugar. Okay, so those are just few. Animal breeding methods. There are a lot of methods in animal breeding. Okay, the first method of breeding is selection. What is selection then? This means choosing parents with desirable genes and rejecting those with undesirable ones. Okay. Breeders also utilize inbreeding. This is a technique in which close